In this video, we are going to discuss about Cosmids. Now, these Cosmid are hybrids of lambda-phage and plasmid. Okay, and their DNA can replicate in a cell like that of plasmid, or their DNA can be packaged like in case of lambda-phage. Okay, so Cosmids are just the hybrid of lambda-phage and your plasmids. Now, Cosmids can carry DNA inserts 3 times in comparison with size with the lambda-phage. Okay, they can carry about 45 kb of the DNA inserts. Okay, 3 times greater as compared to the lambda-phage. Now, the development of this Cosmid vector was based on the observation that 200 base pair DNA sequences in the lambda-phage genome, which are known as Cosmids, are required for lambda DNA packaging and for the formation of phage particles during lytic cycle. Okay, during lytic cycle, the Cosmids are required, okay, to package the DNA to produce the phage particles. Okay, and these Cosmids are about 200 base pair in length. Now, cleavage at Cosmids of by the lambda terminase proteins. Okay, this lambda terminase proteins are able to cleave the Cosmids during phage packaging, and when these proteins, when they cleave the Cosmid, they produces 12 nucleotide cohesive ends at the termini of the linear lambda genome. Okay, so when these Cosmids are cleaved by this lambda terminase protein, then that produces a cohesive end made up of 12 nucleotides at the termini of the linear lambda genome. And the recircularization of this lambda genome after the bacterial infection is facilitated by the base pairing between these complementary cohesive ends. Okay, so when you cut the Cosmids, you produce cohesive ends in the linear lambda genome, and when you want to recircularize this linear lambda genome, then you just base pair between those complementary cohesive ends, and that is how the recircularization of the lambda genome occurs. Now, for in vitro packaging of the DNA into the lambda phage, the only requirement is the presence of these 2 cos ends that are separated by 37 to 51 kb intervening sequence. Okay, for in vitro packaging of the DNA, these 2 cos ends are required. Okay, this is the only requirement. So these 2 cos ends present at the termini of the linear lambda genome are very important for in vitro packaging of the DNA into the lambda phage. Now, this Cosmid is a plasmid that carries Cosmids. Okay, the thing is that if you have Cosmids, then it can be packed as a virus, and this Cosmid is a plasmid which has the Cosmid, so this plasmid becomes able to be packed as a virus. So it increases its efficiency to deliver the DNA. Now, most Cosmids have the size of 5 kb. Okay, 5 kb is the general size of the Cosmids. Now, Cosmid cloning vectors with DNA inserts of about 30 to 45 kb can be packaged in vitro into the lambda phage particles. Okay, so you take Cosmid, you insert a DNA sequence of about 30 to 45 kb in it, and that Cosmid cloning vector then is packaged into lambda phage particles. And after that, what you do is you infect E. coli cells with this Cosmid packaged phage, and then this E. coli supports the autonomous replication of this circular Cosmid plasmid, because the genome of this Cosmid packaged phage contains the COLE1 origin of replication and beta lactamase gene. Okay, in the cos sequence, the cos sequence is linked to both of these sequence, and that is the reason why these E. coli cells support the autonomous replication of circular Cosmid plasmids. So you make a Cosmid, you put it into E. coli cell, and then E. coli cell will replicate that Cosmid. Okay, that is how it is going to work. So this is how a plasmid looks like. Okay, this the size of this DNA sequence, this circular DNA molecule, is about 5 kb, and that is a Cosmid. And the distinguishing feature of this Cosmid is that it have the presence of cos sites, as you can see, and this cos sites enable the vector to be packaged into the bacteriophage head, and that is what is increasing the efficiency of this Cosmid. So yeah, it contains multiple cloning site, ampicillin resistance gene, and origin of replication, as the other vectors do, but other vectors don't contain the cos sites. Okay, other plasmid vectors don't contain cos sites, but this Cosmid also contain cos sites, and that is the distinguishing feature of this Cosmids. Now there is something known as fosmids. Okay, now let's discuss about that. Now these fosmids are based on the bacterial F plasmid, and they contain F plasmid origin of replication, and they also contain lambda cos sites as well. And the thing is that the fosmids are similar to Cosmids, but the copy number of these fosmids is low in E. coli. Okay, these fosmids are able to hold the DNA inserts of about 40 kb in size. Okay, so this was a kind of short description of these fosmids, and that is all for this video. Thanks for watching.